Hello, sir.
Hi, how are you?
I'm fine. How are you?
Good. Susan,
What happened?
what do you mean?
Oh well. Oh well, my screen. Anyway, it's okay.
Okay. And Barshan is here. All right. So, yeah, Utkarsh is here. Barshan, would you like to say something?
So, am I audible right now?
Yes.
Okay. So I wanted to present something last week, but my Gypsy was very buggy, and I was not able to hear anything or open my camera. So I would like to present it today.
Okay, sounds good.
Okay. So I was working on pre-processing the epic data to make it compatible with my model, which is explainable spatiotemporal graphs. So normally the data is not made for working with it on graph neural networks. So what I did was I converted it to NumPy arrays, and I'll brief on it in the later slides. So what we have here is one epic dataset. So I bulk downloaded the entire CSV. We had like around 220 CSV files with different datas. So we have the columns over here, which is the cell, time, none, global, local, plot, and we have the XYZ coordinates, the size, and the gene weight. So what my goal was to fit this into the sensor shape, which is like R to the power n into d into t, where n is the number of cells, d is the number of features, and t is the time step. So I wanted to have a dynamic graph which stores the cells sparsely along the edges across time. So this is my entire a small short flowchart that I made. So we have the raw epic dataset over here, and we are inputting that dataset into my pre-processing pipeline. And we extract the features from there, and we generate a alive mask, which means that it tells us which cells are alive at that moment and which are not. And we also construct the edges, and then we output a NumPy array having the features, this alive mask, and the edges. And then we pass the process data into my model, which I'm going to train on it. So this is the input which I have. So it contains the biological data, which is like ABA, ABAL, and the time, discrete time point, and features like the position, size, and the block, which is the fluorescent signal we have. Now, what does this each.npz file contain? So it contains a feature tensor, which is x, which has n, 5, t, which n is the number of cells, 5 is the number of features, and x, y, z. And I took these 5 features. I can change this along the way, like if I want something else or if the accuracy is not enough, so I can change my features itself. So, and t is the time steps. Now I have alive mask, which is a Boolean function, where true means the cell exists at that time, false means the cell is not yet born or already gone. Now, these are the edge definitions, like to see if that edge exists or not. So this describes an edge, basically define an edge. So the edge source I have, which is like N32 data type, and I have the edge destination. And I've also have the time edge t, which carries the time step of when the edge exists. And other than that, I've also used a mapping function IDX to cell to make it easier for me to go to that cell directly using its identity. Now I have T0, which is the starting time, and T, which is the total time steps, time info of an edge. And source file is also it traces back to the original file or the CSV file. That is also there in the NPZ file. So basically, NPZ is a compressed collection of arrays, and it is a binary form of data, so we can't see it like it's not readable humanly, but you have to use the NumPy library to read it. Now, this example where I have fetched a data from my process data, and I have ran a lot of scripts where I have used each function and I've seen how it returns the value. So I've taken this file cd01 bright.npz, and I have the feature matrix, which is the 688, 5, 210. So 688 means is the number of cells that file has. 5 is the number of feature which you are taking into account. 210 is the number of time steps. And then we run a Boolean alive function, which says how many edges are alive in that function, in that NumPy array. So it's around 20,430. And these are alive edges total at the end, like at the 210 time step. And the total number of edges is 56,605, like for the whole time steps. So I've also ran a Boolean function which asks if the edge ABA to ABAN is true or not. So it does have that edge in the NumPy array or in that CSV file. So it's returning true. And I've also included this, like how many times this edge exists in the whole array. So there's 14 times the edge exists. And so there are multiple ABA to ABAL connections. So that is 14 number of edges. So since I was about to present this last week, this week I also created a small documentation, which I'll also like go through briefly. So let me present that also.
Okay.
So this is like a documentation which I did on my blog. So it's a really detailed documentation, since I didn't want to leave this behind, because this data can be used like in future as well, because this is most adaptable for using in graph neural networks or when we are working with graphs. So this data can be used there also. So I'll just go through a table of contents. So I've went through the what is this data set and a loop in 10 lines, like what the whole feature matrix does. And after that, I also have like in depth, I have went through like this quick start is just like a short info to the whole pipeline. Now, in depth is why this data set matters. So we have this complete lineage, and it talks more about the epic data set and how the thing about C. elegans is it has a lot of single cell data. So it's easier for us to get features from it and work on that. So after that, we have the raw data format. Like I talk about the epic CSV files that what we have, and we have the column breakdown. Then I go through each of the functions that I have in my pipeline with the code. The code is also available over there. So I have entered here, like I have noted down each function, like how it works, and also with the example to it. So these are the functions, like they build the edges, and we have built the spatial edges. Spatial edges depend upon the Euclidean distance, and the linear edges depend upon the connections. And then we have the output specification, like what goes out from the NumPy npz file goes out, and these are all the functions that we have. And I've mentioned this, like we have the typical contents of the file, like what is there, like with a timestamp also when it's shown. And I've also mentioned the data structure of a file, like the number of cells we have and of a single CSV. And other than that, I've also ran a few validations. So we don't have any null values in this dataset. And there is no nodes in edges exist. So there is always a edge which always exists. So that if we work on this dataset, it doesn't run into a error, because no edge exists. So then we have sparsity, like the graphs are sparse and not dense, so it will also be not heavy on the memory. And other than that, we have the linear closure, like parents exist for all daughters. That's what I said, like edges exist. And there were some common issues when I was testing the files. So one of them was like we corrupted or wrong NVZ file, and something about the dense thing. So I didn't have a lot of RAM, so I had to run it one by one. And other than that, these two were also there. We can just go through. And other than that, this dataset is available on my GitHub also. So it's possible I can add it on the DevoWorm dataset file also if required. And that's it mostly.
That's great. It looks great. So this is basically thinking about how you would structure the data set for the model. And then
yes. And make it adaptable.
Yeah. Well, I like that. Yeah. Looks pretty good. Yeah. And then you have all the data. So this is all available on your GitHub site. You put that in the chat, please? for people
Okay,
to
okay.
see. Yeah.
So the GitHub link or the documentation?
You can do both. Be good.
Okay, okay, all right, all right.
Okay. Yeah, so that looks great. I'd have to take a look at it a little bit more to see, what it will probably have that as. Actually, like you and Ashu have been creating these data sets. I'd like to kind of host them on the DevoWorm in our organizational GitHub. So that'd be kind of nice to have. Yeah, so that's the spatiotemporal evolution data set. Thank you, Barshan, for presenting on that.
So I have the raw files as well as the processed one. Like there are two different folders.
Okay. Yeah.
It's easily able to figure, like you can track it. Thank you.
Okay. Yeah. Ashu, did you have something to present today?
Yes, like I don't have any formal presentation to present here because last week I was mostly working on a research paper that I have to publish. So, there is last week I presented my anastamoses table, and a little bit of documentation is that I have created. I want to present that.
Okay.
Okay, this is the documentation of how clusters were formed in that anastamoses table. So basically, I have used the DB scan to form clusters with EPS 15 and a minimum sample of 3. Like DB scan then runs only use two parameters. First, this is the EPS 15 and another is the minimum sample of 3, which makes the maximum distance between cell can be apart and still be considered a spatial neighbor. Like at T53, this process produces 4 valid clusters, each contain 3 to 5 cells. And to identify the switching cell, when the DB scan runs both at both time points, like at T53 and at T54. The results are stored in lookup dictionaries, mapping each cell name to its cluster at T53 and another at T54. For each cell present in both snapshots, we compare its label, at T53 and at T54, the cell labels are compared, and then we can identify the cell that has shifted its cluster from T53 to T54. Like a cell is flagged as a switching cell if 3 conditions are true. Its cluster label changes between the 2 time points. The second one is it was in a valid cluster at T53 and it should be in a valid cluster at the next time point. All these 3 conditions should be followed to consider a cell a switching cell. Like if we talk about the MSPP, at T53 it was in a label 3 and at T54 it was in a label 0. So all the 3 conditions are satisfied. The cluster labels are different and it is present at both time points, at T53 also and at T54 also. And there is one more thing, like a phantom event. Like it is also checked in this, like if the members are, if both the members are similar in both the time slots. Like if in T53, if all the cluster members are same and at T54 also the cluster members are same, then it is considered a phantom event and it is removed from the dataset. And there is one more thing in that table, that we have started from time point 53. Like basically from time point 1 to time point 51, there were the cell present are too few and was spread in the 3D space. So there were no spatial community that has formed at that time point. So we have not considered it. And at time point T52, the embryo reaches the fish first time. The two tight spatial groups appear, but the problem was that to it to be considered, it should be present in the time point just after this one. Like if we are taking a time point 52, then the cell must also be present in the time point 53, and it was not present. Like the cell time point 52 was either in the same cluster or it was a noise in the time point 53. Basically, the pair 53 and 54 is where everything comes together. There are 4 valid spatial communities at T54, T53, and 4 at T54. And the two cells, MSPP and ABALPPP, generally changes their community membership between the two snapshots. So that's why we have started from the 53 time point. These three were the main logic behind that table, and rest there were only the label and the time slot shift.
That's good. Thank you, Ashu. I saw that in you sent it in Slack, so I was looking it over. That's very good work.
Thanks.
And again, I'd like to push that to the DevoWorm organizational GitHub. So yeah, we'll see. I'll probably take care of that. I'll see how I want to have it done. But do you have this on GitHub now, or?
No, sir, I haven't posted anything on GitHub yet.
Okay, yeah. Well, I mean, I could probably do it. In any case, yeah, thank you.
You're welcome, sir.
All right. So that was Barshan and Ashu. Thank you for your secondary data contributions. I don't know if Utkarsh had anything you wanted to present.
Hi, Brad. Hi, everyone. Yeah, I do have a presentation. Yeah, I would like to continue from my previous experiments. I couldn't present last time. I did some work, but I didn't have a presentation at time. I still don't have a well-designed presentation. This is just a collection of my notes from the experiments, so please bear with me. So I pivoted from my original approach, and I would also like to share that plus the results that I've got over these past few weeks. Just to recap, my original plan was to run C302 neural simulations on reticulate connectivity data and extract activity features and apply SINDyG to discover how those features change across development. I faced 3 different problems during this. First, the C. elegans neurons do not generate action potentials because they lack voltage-gated sodium channels. So I fixed that by switching to parameter C0, but then I hit the saturation problem because T1 and T8 stages both had the identical neurons activating at 20 picoampere, like the L1 larval network and the edit network activated identically. So I found a sensitivity window at 2 picoampere where T1 activated 70 neurons and T8 activated 1 single. But the core issue now was that reticulate T1 to T8 are not measurements from the same worm over time. They are 8 different genetically identical individuals, but killed at different ages. But individual worms also differ in about 46% of their connectivity, despite their identical genomes. So when I ran SINDyG equations over it, those describe the population level correlations and were not giving any information about how the nervous system develops for C. elegans. So the problem is that reticulate tells me what the connectome looks like at a stage. It doesn't tell me why. And running SINDyG on the data would give me correlations instead of causes. To understand why the D1 connectome looks the way it does, I think we have 2 things that determine whether 2 neurons would form a synapse. First is the physical proximity. 2 neurons can only connect if their membranes are physically close enough. and synapses are actively forming. Secondly, molecular recognition. Neurons express surface proteins and synapses form when the compatible molecules are present on both sides. Now, both factors must be true simultaneously. So, I initially was considering using SINDyG dataset, which is comprehensive single-cell RNA atlas of all 302 neurons. But it is adult data measured on fully wired adult worm, and the wiring decisions that determine the D1 connectome happens earlier in the embryo. So I shifted to Packard 2019 dataset. It is a lineage resolved single cell atlas of C. elegans embryogenesis. I can extract the gene expression profile of any neuron type at the time point when it terminally differentiates, which is exactly when it's expressing the guidance molecules that would later determine the connectivity. For the gene set, I filtered to 235 genes, 35 curated axon guidance genes known from known pathways such as nectin, FN, WNT signaling, SLIT-ROGO, and plus 200 highest variance genes across neuron types. High variance across neuron types means that these genes are differentially expressed, which is necessary condition for them to encode neuron identity. I'm not using 20,000 genes because that would cause massive overfitting for such small amount of neurons. So the first component is what I'm calling the molecular entity. The model is simple. The probability of a synapse between neuron I and neuron J equals the sigmoid of transport of xi times w times xj, where x is the expression vector that we get from pattern and w is the learned matrix. W in this case is also our NDP, where each entry WAB asks if neuron I expresses gene A and neuron J expresses gene B, and how much does this contribute to the probability that they would connect in future. Should I first share this specific data set, right?
Yeah, this is the so this is Packard data set, okay. And yeah, this is the one from 2019. And this is the things that you described, yeah.
So I will just quickly repeat. The probability of a synapse between neuron I and neuron J equals the sigmoid of transpose of xi times w times xj, where x is the expression vector and w is the learned matrix. And w in my case here is also the NDP, since each entry WAP asks if neuron I expresses gene A and neuron J expresses gene B, and how much does this contribute to the probability that they would connect in future. So if the model learns high positive weights for an entry corresponding to UNC-C, which is a nectin ligand, and UNC-40, which is the nectin receptor, then it would have recovered a known biological X guidance pathway purely from the data without being told which genes matter. So this is my definition for NDP here. And now to prevent overfitting, as you cannot fit 235 by 235 matrix on 131 data points, I implemented W as product of two low rank matrices, U times V transpose, where the rank is 20. This constrained the model to learn 20 most important gene pair interaction axes. And for evaluation, I used the neuron level foldout. I removed all the edges involving a set of held out neurons classes from the training loss. Then evaluated on those neurons in test set. This is stricter than edge level foldout because it still lets the model see both the neurons in training from other edges, and it is essentially interpolation. But neuron holdout meant that the model has never seen these specific neurons in training at all. And if it predicted that their connectivity correctly, it has learned a transferable rule, which is which is my hypothesis. Now, here are the results. On 131 neurons with 468 positive synaptic connections, the molecular model achieved AUC-PR of 0.249 compared to degree baseline 0.227, and this proved like the molecular identity was beating pure topology. The degree baseline used only the information on how many connections each neuron had, it predicted that high-degree neurons connected to other high-degree neurons. This is like the strong baseline because the hub structure is real. I found that during my C302 experiments also. But the molecular model adds specificity. It predicts which patterns a neuron chooses, not just how many. One thing that might look like a negative result is that combined model, molecular plus spatial distance, performs worse than the molecular alone at every tau setting. This is actually a positive finding because when I look deeper into the Packer expression data, it implicitly encodes the spatial organization also, because in C. elegans, neurons that share same embryonic lineage are thus and thus similar expression profiles also tend to be spatially adjacent. So the W matrix which I had was already capturing the proximity to the correlated expression, and thus adding explicit distance added correlated rather than complementary information.
This is experiment 1+2. So this is
These were regarding like testing the trained model, the DGV.
okay.
I used like the Packer for that. So I was like trying to see what the W would actually represent. So the top entries included EFN4, which is an Ephraim family guidance molecule, paired with NRX1, which is neurorexin, a core synaptic adhesion molecule, and NID1 paired with VAB1, MIG5 paired with itself. So W could partially recover real biology. Two gene pairs are reproducible across 20 independent training runs with different seeds, different random seeds, UNC5 and UNC30. Is it changing right now?
Yeah.
Yeah. So the second component is the hypergraph. In Vitulate, each synapse entry has a pre-field, which is one neuron name, and the post field, which is a list of 1, 2, 3, 4 neurons in it. It reflects that one presynaptic active zone can simultaneously release neurotransmitter onto multiple postsynaptic partners from a single release event. So what I found when I extracted these, that 83 to 90% of all the synapses in Vitulate are polyadic across every developmental stage. Thus, the standard pairwise graph analysis would misinterpret the actual connectivity by flattening these into independent edges, which is why we need hypergraphs. 63 specific polyadic configuration were present in all of the 8 stages from Vitulate, from hatching to adulthood. These are established embryonically and never rewired. The most frequent is the RIAR driving RMDDR and RMDVL simultaneously. The right RIA interneuron simultaneously coordinating with the dorsal right and the ventral left ring motor neurons. 42 of these 63 stable hyperedges connected the neurons in 2 different circuit modules. Presynaptic neurons simultaneously driving targets to the in the olfactory circuit and the locomotion circuit, for example. I have yet to dive deeper into this before I can truly understand how I'll be integrating this with my network. For now, I have the temporal DGNN, which is basically GCRN, which I'm using. It is the third component that predicts how the connectome would evolve from a stage to a stage. Is my slide visible?
Yes. Yes.
The node features here combine the static Packer expression, which stays same essentially during all the stages for a neuron, capturing its fixed molecular identity. And I also had the dynamical structural features that change per stage, for example, the in-degree, out-degree, and reciprocity. The graph encoder uses directed sage instead of the standard symmetric GCN. It was important because presynaptic and postsynaptic roles are fundamentally different. A standard symmetric GCN would treat the neurons driving you identically to the neurons that you drive. On the contrary, the directed sage separately aggregates from the incoming neighbors who sends to me and the outgoing neighbors who I send to, and then concatenates them. The temporal component is a GRU, It takes the sequence of node embeddings across stages P1 through D6 and accumulates a developmental history in its hidden state. The decoder concatenates four things for each candidate edge: the embedding of the source, the embedding of the target, their element-wise product, which captures the interaction, and their absolute difference, which captures the asymmetry. This goes through two-layer MLP to produce a connection probability. Now, one thing to note is that I use synapse weights from Wikilead during message passing as log of size times post weights rather than treating all edges as binary. This meant a stronger synapse between two neurons influences their embeddings more during training. Is my screen. Okay, so here were the results from DGNM. The model achieved an AUC ROC of 0.927, predicting D7 connectivity, and 0.937 on held out T8 adult test. I also ran a ablation study to further understand where the performance was coming from and to check, to validate my model. A simple MLP using only the expression features from Packer achieved 0.869 ACROC. This meant that the Packer molecular identity carried important developmental signals. When you add the graph encoding, it boosts up to 0.922, and adding GRU increase it to 0.927. On the subset of 131 neurons that all have Packer expression datasets, excluding the motor neurons, which I'm using zero vector defaults, the AUCPR rose from 0.481 to 0.65 because they were the motor neurons were currently diluting my global model. The most important number is the new synapse prediction result. I specifically evaluated the model only on the connections that didn't exist at D6 but appear for the first time on D7 to validate whether the model is predicting which new synapses form and not just existing connections that persist. So the persistence baseline is at random chance 0.02. The model achieved 0.125, which is 6 times above random. So it is not just memorizing the existing connectivity, which is what I was trying to validate. And I also verified whether there was a leakage. I retrained the model without the D6 in training or evaluation at all, and the AP changed by 0.0009 or 9 to 12, 9 to 10 minus 4. Then I ran a few more validation tests. I ran 12 different validation tests to stress the model. I will just focus on the most important ones. The strongest positive result was the generalization test. I took the AWC olfactory neuron class, AWCL and AWCR, and removed all of their edges from their training loss. The model had to predict the AWC connectivity using only the expression profile of AWC and the expression profiles of the candidate partners. It achieved 0.963 AUC-ROC. The model could correctly identify that AWC connects to AIA, AIB, and AFP, the canonical olfactory circuit, without ever being trained on those connections. The configuration model test randomized all the edges while preserving the degree distribution, destroying all the edge identity, but keeping the topology intact. The GCRing then collapsed to 0.753 AUC. This meant that the model cannot build meaningful embeddings from randomized connectivity, even with the expression features. The 0.174 AUC-ROC gap between the config model and the real data comes entirely from the actual wiring patterns and not the degree statistics. There is one honest limitation. The generalization test, when I ran the generalization test, it also revealed a boundary condition. The ASI and the ASH, which are the two peripheral chemosensory neurons, showed near chance holdout performance at 0.59 and 0.58 AUC-ROC. The model basically fails for neurons that are sparsely connected and have unique molecular profiles and are connected to less central targets. So the model is effectively learning what popular interneuron hub targets accept as presynaptic input. For neurons like AWC, whose targets are well-known hub interneurons, that's sufficient. But for like you see ASI and ASH, whose targets are more peripheral, it's not enough. And also the bilateral neurons also get near identical prediction with correlation of 0.997 between left and right because they have the identical expression vectors by construction. So right now the model cannot distinguish between the AWCL specific from AWCR specific connections. So basically right now the motor neurons are missing and the left and right asymmetry is unresolved and the gap junctions are also not included. I will start by figuring out the left and right asymmetry first. Then I'll move ahead. Yeah, this is it.
Okay. Oh, is that it?
Yes, right.
Okay, yeah. Thank you, Utkarsh. Yeah, it's great. A lot of work there. I like that you were able to go back and explore some of these data sets. So the SINDyG data set, of course, is a molecular data set. And you improved upon that by going to the Packer 2019, which is like a spatiotemporally specific gene expression data set.
Right.
How did you find that to work with? I'm just curious.
Actually, my the basic thought process was the time, the SINDyG captures the adult period, and whereas I was trying to formulate the reasoning behind why the connectomes form the way they do. So I figured that Packer would be better, or basically the one that I can use here.
Yeah. You have the data available somewhere?
Yeah, I do.
Okay. Yeah, could you share the link? I'm just curious.
All right. Actually, it is not uploaded, so I'll just push it to GitHub and then share the link on Slack maybe.
Okay, yeah, it's good too. Yeah, it's fine.
That is fine.
Yeah. Yeah, a lot of good experiments there. Yeah, it's we usually work with cellular data, which is informative, but of course we have the molecular data as well. Which if you don't know how to work with it, or if you don't know what you're looking for, or how to structure it, it can be quite hard to gain any insight out of. This is true, I think, in general. People take molecular data set and they'll get really excited because they have all these genes and maybe all these states if you're looking at gene expression data, and they put it into a plot and they show that there's this maybe some sort of pattern and it's hard to know what it means. So that's always a problem with molecular data, and then linking it to the cellular data. And C. elegans, that's somewhat easy because C. elegans has this sort of deterministic form of development where they're the cells. If you have a lineage, you know what those cells are going to be at the end, and so you can find all sorts of developmental precursors in the data. You can find all sorts of switches in the data that switch from one cell type to another, and it's quite easy. But when you're working with something like mammalian data, let's say for example, it's much harder to figure out what the molecular data actually mean. And we did a paper 2 years ago now, where we were looking across species at different proteins, their paralogs, which are basically copies of a gene that are different in different species, and their activity relevant to or relative to development. So it was and it's kind of hard to draw any conclusions from the data because it's kind of you see a difference and you say, okay, there it is. There's a difference between species. But what does it mean? I don't know. So it's always kind of difficult sometimes to make those links between the molecular data and the phenotypic data, which are the cells. but I think you did a pretty good job here. Having that spatiotemporal temporal resolution helps because, again, it maps pretty cleanly to the embryo and the different positions of the cells. So again, in C. elegans, you have that spatiotemporal resolution that's sort of inherently in the network structure. So in other words, if I take a network of cells and I build a network, and I have this spatiotemporal gene expression data, it links right into that topology. I don't have to do anything to kind of infer what something means. It's a very clear mapping. So yeah, it's very good.
I understood like what you're trying to say.
Yeah.
So like back to this cellular data set, and I'm trying to like map it as if it is molecular one.
Yeah. Yeah.
With the W matrix. I don't have very clear understanding about that right now. Yeah, but I would like to explore it further.
Yeah. Yeah, I think that's useful. Definitely. And and probably paper worthy. I mean, building a molecular model of this, like at this level, it's pretty good. Going to be useful. All right, thank you, Utkarsh.
Thanks a lot.
Yeah. Did anyone else want to present? Susan or Morgan or Jesse?
No, I don't have anything. I've been sorting papers, so hopefully something will come of that, but
Yeah.
not today. Yeah.
Okay. Well, thanks. Okay. Yeah. All right. So I have two papers. This one is from 2024. These are both on C. elegans and talking about C. elegans modeling. This one is from the bioarchive from the biophysics archive. And the paper title was called A Markovian Dynamics for C. elegans Behavior Across Scales. And so the abstract here reads, how do we capture the breadth of behavior in animal movement from rapid body twitches to aging? So rapid body twitches are things that are happening in the embryo when you start to get muscle. What tends to happen is you get the muscle cells first, and those muscle cells start to twitch because the muscle is tonically active in the embryo, and then the connections from the connectome start to come and innervate the muscle. And then eventually, when the worm is hatched, it starts to have these movements where the activity of the muscle due to something the connectome sending it. So it's basically goal-directed behavior. It becomes part of this motor circuit. But you do get these rapid body twitches before that, and so this is something that's people have looked at. It's something you can get out of the data if you have nice high resolution microscopy images of the embryo, and you can see the muscle cells and they're twitching and they're sort of getting ready to get hooked up to the connectome, and then right after it's hooked up, then it can move the worm. So that's basically what we have. Not to mention that the worm has to undergo movements and generate forces to hatch out of the egg. So that's another thing that's going on there. But this is kind of at the beginning of development. And then, of course, we also want to look at aging, which is where you get differences in aging. As the worm ages, of course, we have the larval stages, and then we have the adult stage. And then there's a stage kind of at the end of the worm's life where they slow down and eventually die. And so this is something, and of course, it's also reproduction where the worm is laying eggs and things like that. So these are all different kind of life history stages of movement. And we want to capture all of this behavior because we tend to think of these circuits in the connectome as generating very simple movements, very stereotypical movements. But in fact, there's a lot of movement behavior diversity within the life cycle of C. elegans. So in this paper, they use high-resolution videos of the nematode C. elegans. We show that single dynamics connects posture scale fluctuations with trajectory diffusion and longer-lived behavioral states. So this is where you get these fluctuations in sort of the what they call the posture. And if I know most of you probably don't know anything about the work that's been done on sort of capturing C. elegans movements. There's a data set that OpenWorm hosts. It's the movement database, and I'm not sure about what the status of that is right now, but basically it's a database where they've taken worms in culture and they've taken images of the worms, of their posture, meaning that they've taken pictures of the worms in different poses. And then they can infer movements from that. So we have these stereotypical movements. If the worm is in liquidy medium, they'll wiggle really fast to sort of swim. If they're on a gel, or if they're on some sort of substrate that's more solid, they'll kind of slither, and that's their mode of movement. And then they also have these quiescent states where they're just still. They don't make any movements. And so you can observe all of these, and then they can move forwards and backwards and things like that. So you want to be able to extract those postural sequences or those postural states, and then build a model that shows kind of the possibilities for movement. So in this case, they take short posture sequences as an instantaneous behavior measure, fixing the sequence length for maximal prediction. Within the space of posture sequences, we construct a fine-scale maximum entropy partition so that transitions among microstates define a high-fidelity Markov model, which we also use as a means of principled coarse graining. So they're building this model of basically of states with transitions. And you can have these different states that are postural, and then you observe how they transition from one posture to another. That's basically what they're doing. Now, the OpenWorm group, they published a paper a long time ago now. It's probably been about 10 years, called eigenworms, they call them eigenworms because they use eigenvalues and eigenvectors to do this kind of prediction. So, there are different kind of methods for doing this. So this is the high fidelity Markov model that they're using here. They're using this as coarse graining, or a coarse grained model of movement. We translate these dynamics into movement using resistive force theory, capturing the statistical properties of foraging trajectories. Predictive across scales, we leverage the longest lived eigenvectors of the inferred Markov chain to perform a top-down subdivision of the worm's foraging behavior, revealing both runs and pirouettes, as well as previously uncharacterized finer scale behaviors. So they're actually using eigenvectors of the Markov chain here too. So they have this sort of, they're working from sort of this high dimensional data, and they're distilling it into these different mathematical constructs. So they're able to reveal different types of behaviors, behavioral sequences, etc. So, there's a temptation to think, of course, that the movement behaviors are quite simple, but in fact, if you look over time and you reconstruct some of these sequences, you'll see these different types of finer grained behaviors. We use our model to investigate the relevance of these fine-scale behaviors for foraging success, recovering a trade-off between local and global search strategies. So this is what they're doing. So they kind of point out that, let's see. So the introduction is, from molecular motors contracting muscles to neurons processing an ever-changing environment, where the large-scale diffusion of hormones or other neuromodulatory chemicals, animal behavior arises from biological activity across innumerable spatial and temporal scales. So this is a spatiotemporal aspect. With an instantaneous snapshot of all these variables, the future behavioral states of the animal would be uniquely defined, a biological setting for Laplace's demon. Of course, such an approach is practically unrealizable. We are limited to a much smaller set of observations, and the unobserved degrees of freedom will generally induce non-Markovinity. So this is where you have this, that your biological system, you may make the assumption that it's sort of a Markov process or a Markovian process. And so, there's this sort of balance between, kind of having this sort of Markovinity available to you to do modeling of states, and this non-Markovinity, which is where things are not as sort of randomly distributed as you would like. In animal behavior, interpretations of this memory guide our understanding of the complexity of the process. But what if we could use our observations to construct memory-full state variables that admit predictive yet minimal memory dynamics? So that's what they're doing here. And they're doing this with movement data. Here we see such a Markov dynamics from the time series of posture in the foraging behavior of C. elegans. And so this is where let's see if I can find a figure. This is a figure here where this is maximally predictive posture sequences reveal the space of short time behaviors. So here they have this worm posture, which is just the worm sort of sitting in this state. They extract this sort of time sequence, or they infer the time sequence, and they show these different components of the movement sequence. So a forward wave, a reversal wave, and a turn. And then they have this where they kind of calculate this out from these images. So different postures at different time points, and then just try to infer the movement from those discrete observations. This is where you have this analysis of the postures in space and in time, and then you have these are these maps. I'm not sure exactly what they are, but they basically measure points on the body and their movement. So you see this sort of the body wave velocity. So a lot of the worm movements are oscillatory. So you're getting these cycles per second. And then you have a total curvature of the body in radians. So you see that this part of the worm, you have these different markers here, or these different points. And between postures, they'll move around. They'll cycle, so they'll exhibit oscillatory behavior, and they'll exhibit curvature behaviors, which are in radians and described in this sort of visualization plot. And you can see the heat map here. So that's kind of what they're doing. And then they're extracting these eigenworms. So they have this set of eigenworms where they obtain a 5-dimensional eigenworm time series, and that's how they build these models. So this looks like a pretty good paper. If you're interested in C. elegans movement dynamics, I know we don't talk about that a lot in this group, but it's an important point. And of course, in development, you're going to have different movements than in the adult. The second paper is from eLife. This is, actually, it's not a new paper at all. It's from 2016, but this is something that we haven't talked about in the group. This is a stochastic neuronal model that predicts random search behaviors at multiple spatial scales in C. elegans. So this is, let's look at the abstract of this paper. There's some pretty big names in the field on this paper. Eduardo Izquierdo, Cornelio Bargman, Sean Lockery, people you'd be familiar with if you know C. elegans biology. And so the abstract reads, random search is a behavioral strategy used by organisms, from bacteria to humans, to locate food that is randomly distributed and undetectable at a distance. We investigated this behavior in the nematode C. elegans, an organism with a small, well-described nervous system. Here we formulate a mathematical model of random search. So this is where they're searching for food and they're moving towards the food, but you actually have this spatiotemporal model that is external to the worm anatomy, and it's describing its movement in space. And so they're formulating a mathematical model of random search, abstracted from the C. elegans connectome, and fit to a large-scale kinematic analysis of C. elegans behavior at submicron resolution. They're actually using the connectome to build a model of random search. In other words, the ways in which the C. elegans can move towards food sources, and it constrains the model, and then you have this kinematic analysis of behavior. The model predicts behavioral effects on the neuronal ablations and genetic perturbations, as well as unexpected aspects of wild-type behavior. Wild-type behavior being the worms that are sort of default or wild-type genetic strain, as opposed to mutant strains that exhibit different types of behavioral deficits or other types of behavioral competencies that you might expect from sort of a standard C. elegans. And so the predictive success of the model indicates that random search in C. elegans can be understood in terms of a neuronal flip-flop circuit. So they actually propose a kind of a different type of circuit, or a specialized type of circuit, called a flip-flop circuit, involving reciprocal inhibition between two populations of stochastic neurons. Our findings establish a unified theoretical framework for understanding C. elegans locomotion and a testable neuronal model of random search that can be applied to other organisms. So here they're thinking about movement, not just as sort of the position of the worm and then inferring different types of movements from those dynamics, but actually understand what the worm is doing and where the worm is moving towards, and maybe some of the decision making that's happening as a result. And so this is kind of an interesting model because it actually proposes a circuit type that needs to exist. And so there's the switching that has to exist in the circuit, and that controls sort of the movement to different places. So, a lot of times, we think about organisms' foraging strategies. That is, what are the different ways they use to acquire food? And so this is something that, of course, random search, you observe this in bacteria, you observe this in all sorts of single-cell organisms. And this is, we kind of think of decision-making as being this sort of very detailed sort of search criterion, but sometimes random search works just as well as a foraging strategy. And so that's what they're proposing here. And so some of these, embedded in this foraging strategy are different types of movements that the worm has to make. So, they talk here about how, you have this random search strategy, but you also have these constraints of movement of the worm. So rather than attempting to exhaustively search a region of interest, the organism samples the environment at randomly selected points. This is achieved by executing a series of straight line movements called runs. So they're running at these random points, terminating at random intervals by sampling episodes during which the organism may or may not find prey. So they have a certain success rate associated with these runs. Sampling ends in a reorientation event called a turn, such as the next run is usually in a different direction from the preceding one. So they make a run, they turn, and then they make another run, and they just basically transect their environment randomly for food, and they have a certain success rate. In optimal random foraging strategies, the probability distribution of run length is matched to the statistical distribution of isolated food patches or pre-items, with power law distributions predominating. So they actually can model this using power laws and thinking about like what's the most efficient random search strategy. In other words, you might have very long runs that punctuate a bunch of different short runs that the worm might make. So the worm makes a bunch of short runs and then makes a long sweep through the space, and then another bunch of short runs and another long sweep through the space. And that's distributed according to a power law. So these are different things that people have observed in a wide range of species, and it's called random search, but they don't exhaustively search their environment. They have some sort of strategy. It just tends to be at random. And so, despite the universality of random search, what little is known about its neuronal basis, in part because of the difficulty of recording and manipulating activity in the brain of an unrestrained animal while it explores a large region of space. So that's why they're doing this in C. elegans. And C. elegans serves as a good model for this because you have these relatively simple movements, a relatively simple connectome, and you can connect all of this together. So this is kind of just summarizing what they did in the paper. They measure this worm track, which is like the worm moving from one point to another in a culture. So this is a two-dimensional space that they're navigating, and you can see that they're making these random movements. It looks like a directed Brownian motion is basically the way to describe it. So it starts here and it ends here, and it kind of does this Brownian motion track, and it ends up in some direction, but it doesn't go in a straight line. It just kind of explores around that path. Sometimes they diverge and kind of wander around, but they always make it over in this direction. So they're just exploring the environment for food. They have no idea where the food is. They just have to make these runs and hope for the best. This is the speed distribution, so you can see that the speed, there's a slight increase in speed and then a decrease at the beginning of the run, and then speed increases in the middle of the run and then decreases towards the end. And then they characterize us with sort of this distribution of speed over time, or I guess across the trajectory, millimeters per second. And then you have this autocovariance, which is just looking at overruns, different runs, and across the run. How does it correlate? And then you have this heading change, which means in what direction in terms of degrees is the worm headed. And you see that over time, the heading change is quite variable, much more variable than at the beginning of a run. And then you have these movement state transitions. So they're doing this sort of a similar thing they did in the last paper, but they're looking at these longer term behavioral states where they're making transitions between the different states. So you have this Markovian model basically of movement states that are, the worm is transitioning between these movement states, and you can extract that from the trace of this movement trajectory over time. So you see that they can identify these state transitions as points where there's a shift in velocity. So you can see that there are these dips in the velocity, and they kind of, or these rises in velocity locally, in a local part of this trace, this time series, and those are the state transitions that they've identified. It's a little bit different approach than the last paper. And then you have these velocity distributions. It's a probability density function of the different velocity distributions. This is in millimeters per second. You have these different transitions, including the time after transition. So you're looking at different transitions and their aftermath. And then you have this dwell time distribution, which is where they measure sort of the pause or the duration of a pause. So when they're making these runs, they're not sort of uniform. The point is that they're non-uniform, that they speed up and they slow down, and sometimes they stop and search locally for things. You see this little cluster of points here in the middle. That's where they're stopping, maybe searching locally for things along that run. So this just goes to show you that you don't have this. It's not a deterministic process in a lot of ways. and they're able to find food with this random process given structure. So this is all dependent on the connectome, and then they talk about the stochastic switch model, which is where they propose this minimal model for the control of random search behavior, where you have these two opposing neuron-like units that can exist in 4 distinct states, corresponding to forward locomotion, reverse locomotion, and then 2 pause states. So when we looked at the pause data or the dwell time data, that's those pause states. So they reverse direction, and they have to switch between forward and reverse locomotion. And then they have these 2 pause states that they have to switch to during this run. So that's what they're modeling here. And so this model differs from a previous model that represents the worm as a point in shape space, in that here velocity is measured directly by observing the motion of a point on the body surface relative to the substrate, rather than indirectly by the temporal progression of shape changes. So in the last paper, they kind of took that approach where they looked at the shape changes of posture, and they kind of inferred movement behavior from that. In this case, what they're looking at is they're looking at a point or they're observing the motion of a point on the body surface relative to the substrate. So they're taking the substrate and the body point on the body surface into account. So this is a little bit different way of doing this. And so, this means that they're not modeling a Markov process directly, as with the case in the last paper. They're actually looking at this as a time series, as a dynamical system, more or less. And they're kind of getting to that same thing that they were getting at the last paper in a different way. So I think that's all for today. Did we have any questions or comments? Okay, Barshan?
I remember presenting the first paper in the month of January, I think.
Okay.
I started the meeting with that paper, yes.
Oh, okay.
Yeah.
Well, I wanted to go over it again.
Yeah.
Right.
That's it.
Well, that's good. All right. All right. Thank you. Have a good week.